Good evening, everyone. My name is Christine Jankowski, and I'm the archivist at the Lloyd Library Museum here in Cincinnati, Ohio. Thank you all for joining us tonight for our program, The Chemistry of Botanical Beverages, From Sweet to Bitter, with Professor Edward J. Kennelly of Lehman College, City University of New York, or CUNY. This presentation is part of our programming series for our current exhibit, The Botany of Beverages, where we show and talk about the plants found in some of the world's most beloved beverages. We also showcase the methods used to create them, such as pasteurization and distillation. Library co-founder John Uri Lloyd used some of these methods with native plants to create the products for the Lloyd Brothers line, which further shows that beverages exist at the intersections of science, medicine, commerce, and culture. Professor Kennelly's research has centered on natural products from food and medicinal plants that impact human health. He has published over 170 peer-reviewed papers in the field of natural product chemistry and has held research appointments with the FDA and international universities, including Minzu University of China. He's been a professor at CUNY for over 25 years and is the current chair of the Department of Biological Sciences. He's an honorary member of the American Society of Pharmacognosy, as well as their past president and the former editor-in-chief of their newsletter, which is how I first met him. Tonight, Ed will take us through some of his research on the plant chemistry that gives certain botanicals their characteristic sweet or bitter flavors. Let's give a warm welcome to Professor Ed Kennelly. Thank you so much, Ed.
Thank you very much, Christine. It's really a pleasure to be here at the Lloyd and virtually, and really this opportunity to talk about my research. I've interacted with the Lloyd since I was a graduate student at Washington University in St. Louis. I remember coming to the Lloyd in Cincinnati in about 1990, looking for more information about a medicinal plant I was working on from Jamaica at that time. And the staff in the library was just so helpful for me as a graduate student back then. And throughout my career, I've maintained interactions with Lloyd. Christine and I work on a newsletter together for the American Society of Pharmacognosy. And I've had many other opportunities to visit the Lloyd. And it's just a wonderful resource for those of you in the audience who haven't gone to the Lloyd. I really hope that you have a chance to go and visit and to see this exhibit or other exhibits that they have, and also just the tremendous resources, both the books and archives that they maintain at the library. So thank you very much for this opportunity. It's really an honor. It feels like an institution I've had a long history with, and I'm really very happy to come and speak to you about some of my research. When Christine and others at the library asked me to speak about beverages, I had to stop and think about my career, and it brought me down memory lane. I realized at this point that many of the plants I've worked on really are parts of beverages. And I hadn't really thought of that focus before this lecture. So I kind of try to restructure some of the things I've talked about in the past in a way that is really kind of beverage oriented for tonight's talk. So let me share my screen with you. So tonight I'll be talking about three different plants that we've worked on over the years. But first, I'll give you an introductory. For those of you who are not as familiar with plant chemistry, I'll try to introduce some basic concepts about plant chemistry to you, and then talk about three examples of plants that are used throughout the world as beverages. Some of these you may be very familiar with, some of these less familiar with, and I hope these are ones that we've studied intensely in our laboratory chemically, and I'll try to give you an indication why that's been important in some of the findings that we've gotten from these three plants. The one is Ilex guayusa, which is a South American holly. That was some work I had started when I was a graduate student. Then we have Merceria vexator, which is Joba tacaba, and finally Humulus lupulus hop, which many people are aware of because of its use in beer. And then I'll give you some conclusions and some future directions that I see in botanical beverages. So I like to think of plants as small chemical factories. Plants, as we know, are not able to move, so they have to respond to their environment. They have evolved to respond to their environment in innovative ways, ways that mammals and other organisms have not had to do. And one of those ways is through their chemistry. They're able to respond to their environment, by producing certain compounds. There are compounds that are fundamental for life. This will happen in all of us, from humans to plants to bacteria. This is what we call primary metabolism. I think we can think about these as proteins or carbohydrates or lipids. These are molecules that are necessary for all living organisms. Organisms cannot survive without these compounds. But in addition to these compounds, plants and certain other organisms also produce a huge array of compounds that are really to make it more fit for survival in its environment. These we call often in plant chemistry, we call it secondary metabolites. These are compounds that are produced by plants, and they're used either to defend the plant from biotic stress or abiotic stress. These compounds are also known, you'll hear me talk about them as secondary metabolites, phytochemicals, or natural products. I'll use those terms interchangeably. These are basically a whole array and whole arsenal of compounds that plants produce as a way in which to be uh, better adapted to their environment. We think about a plant, a simple plant like a mint plant is producing many different compounds. And it's important that when we think about the plant, is to review the nomenclature of a plant like uh, all plants have a genus and a species. This was developed by Linnaeus, a Swedish botanist, several hundred years ago when he developed this binomial system. This is important because it helps us to uniquely identify a plant. We might call it white peppermint, but one person might call white peppermint may be different from what another person calls white peppermint. So the way in which the taxonomy of the plant is best classified is using this Latin binomial, not by using common names. But tonight's lecture, I'll interchange common names and Latin binomials at times. But just be aware that I think the greatest precision comes with the Latin binomial. When we think about chemistry, the chemistry is often related to certain plants have evolved in certain environments to produce certain types of natural products. So we'll see that, often if you're looking for error, we know through probably our culinary experience that, a lot of mint-related plants are going to produce a lot of aromatic compounds. Those are produced by a specific type of biochemical pathway that are present in many plants, but especially in the mint family. We get in especially many different species of related species produce these similar types of natural products. So if we're looking for natural products, one way we might think of it is called chemotaxonomy. We look to see what other plants produce that compound, and we think that if they're more closely related to the plant, the more likely it is that they may express some of these similar types of natural products. So plants are really chemical factories, and I'm always curious to see, and I think we have a pretty general audience tonight, if you can imagine how many unique compounds that a mint leaf might produce. So I think, Emily, do you have a poll there for us to fill out? Yes, there's a poll. If you scroll down on the right-hand side, you can answer the poll question. Do you think a mint leaf produces tens of compounds, hundreds of compounds, thousands of compounds, or millions of compounds? If you wouldn't mind just submitting your answer, and we can get maybe. I don't think I can see that, Emily. Maybe you will be able to give us the results in them. Oh, thank you. Well, okay, so we have uh, most people think it's thousands of compounds. I agree with you. And I think I did ask AI just before the lecture how many AI thought. It thought hundreds of compounds. I don't agree with AI. I think I agree with the audience tonight that most plants would be producing thousands of compounds, perhaps even up to 10,000 compounds or more. We Part of our job as plant chemists is really to understand this really amazing plant diversity, this chemical diversity in plants. And that's what we've developed a lot of different tools to look at that over the years. And I'm going to talk about projects that I did 35 years ago and projects that we've done more recently. And the tools have continued to improve, and our ability to see these thousands of compounds and to identify these thousands of compounds has really improved dramatically over the years. So I'll go through some of that in tonight's lecture as well. So as I mentioned, plants, why do plants produce so many chemicals? It's not for humans. We enjoy their taste in different drinks, or we have adapted them for many foods and drinks. And we've many people are attracted to these. Uh, but really, the plants are making them for their own uh, um, benefit, for either for defense. So we can think of, caffeine as also serving as a defense compound besides being a stimulant for humans. Um, it could be for attraction. So some of the sweet compounds or some of the other compounds may be rewards, the flower scents or other things for bees. You know, some of the natural products, a lot of the flavonoids are used for protection against UV stress. And then it can also, some of these compounds are used also for adaptation uh, in uh, like the milkweed and caterpillars. So there's many reasons that plants are producing it, but it's really for their benefit, for their fitness, for their evolutionary adaptation to an environment or to, perform better in a certain situation. Um, so I'm going to start my kind of look at three different plants. The first plants I'm going to talk about that we worked on were really fall into the group of stimulants. These are stimulating beverages, and we know that there's many different stimulating beverages, and they can all uh, trace their origin to primarily one specific compound, a methylxanthine known as caffeine. Caffeine is found in many different stimulating beverages. And the exhibit at the Lloyd has a very nice section, if you have seen the exhibit, where they talk about many of the different caffeine containing plants. Some of these plants are very familiar to us. Let me get my pointer here. We know chocolate cacao produces caffeine, obviously coffee as well. But beyond that, there's many other plants that have evolved to produce caffeine as well. Besides coffee, we have tea, chocolate. Yerba mate, if you're familiar with the drink of these Argentinian gauchos, this is you'll be familiar with that. Guarana, and also the cola nut. So these are all examples of caffeine-containing plants. What's interesting evolutionarily about the caffeine-containing plants is that they aren't all clustered closely. I talked about chemotaxonomy before, so you would imagine, well, maybe if I'm looking for caffeine, I should look in a very specific group of plants that evolved to produce caffeine, because we know not all plants produce caffeine. Your hypothesis might be, well, maybe it has to be closer related plants that produce caffeine. That's actually not the case. And this is a case of convergent evolution, that we know that this caffeine has evolved independently multiple times. And this has happened, this is a, evolutionary tree showing some of the major uh, uh, plants, genera of plants here. And we can see that over time, we can see that uh, the caffeine has evolved independently. So from chocolate, which is not at all closely related to uh, um, coffee, but they independently have evolved to produce caffeine. So this is an example in which we can see that an important phytochemical can be evolved to be produced multiple times from different species. So the one caffeine-containing plant I want to talk about was really one of the first projects I did as a scientist in the laboratory of my PhD mentor, Walter and Memory Lewis at Washington University in St. Louis. I had the chance in 1988 to go with them to South America. We went to the Amazon of South America. We were in Ecuador, and we were in a region pretty much at the border with Peru uh, in in the Amazon region there. And one of the plants that they use among the in that region is called Ilex guayusa. So Ilex guayusa is also like yerba mate. I mentioned yerba mate. That was the drink of the gauchos. This is a different species. This is Ilex guayusa. Ilex guayusa is used in the Amazon. It is in the holly family, and it is a shrub. So you can see the leaves similar size to the hollies that we would see in North America, but not as toothed as those hollies. And they can contain caffeine. They can contain 1.7 to 3.48% caffeine. Uh, and these have been, uh, this is a well-known and well-studied plant because it's widely used in this region as a stimulant. What the Lewises had observed, they had made multiple trips to this to work with the Jivaro or Shuar people of South America, and it is a well-known ritual that the Shuar people have that early in the morning, that somewhere around 5 o'clock in the morning, that people will wake up and they drink large amounts of guayusa. So not just a cup or two, they're drinking loads, many, many cups of guayusa. And after that, they start to vomit, right? So there's this vomiting. It's a ritualistic vomiting ceremony that happens. Uh, uh, it's pretty surprising, but it's something that the Lewises observed, and many other people had observed among this group of Jivaro people in the Amazon. The question the Lewises had, and as a doctoral student, I became interested in is, does this plant contain other compounds that might cause people to vomit? So there's a whole group of compounds that are known as emetic compounds. Was this plant producing some kind of emetic compound? And we analyzed by HPLC, and we did some analytical chemistry, and we found that this emetic was there was no emetic compound that was found in the plant. So the plant was not containing emetine or other well-known emetic compounds, but it was likely that the people were simply overdosing or drinking high levels of this caffeinated beverage, and um, then through learned processes they would vomit out the excess guayusa. It's part of the culture. And it seems to be, been done for, many years. And, the Lewises even show that young people were taught how to vomit using a feather. So it was certainly a reaction that people were taught over time. So this caffeinated plant was being used by this group in the Amazon, you know, apparently as a stimulant, but it also, we know that high levels of caffeine can actually have many negative effects. So I think, we hypothesize that they actually are vomiting out the excess caffeine. Uh, I guess the question would be why not just drink less? And that, we couldn't really answer that. I think it's part of the culture that they really have learned to have this ceremony and um, really to um, um, make this part of their morning ritual. If you want to read more about this, this is a paper that we wrote back in 1991 from our work with the Hevero. This is the ritualistic use of holly that we published in the Journal of Ethnopharmacology. And this is Walter and Memory Lewis, my PhD mentors, who were the really the impetus for this study. When I started my own research lab, I really became an interest in some of the health benefits of plants, especially whether botanical beverages could help to cure or prevent diseases. As we did my early work, I didn't have a budget to go to South America, so a lot of my work was done in South Florida, and a lot of the plants I began my career with were plants that were at least grown in South Florida. We knew well that beverages had many positive health effects. Tea, besides being a caffeine-containing plant, tea is also important because it contains many different polyphenolic compounds like EGCG, right? So these are polyphenolic compounds. These are known to be involved in many positive health effects. They're important antioxidants. They're important as far as cancer prevention is concerned. So there's been a lot of epidemiological studies that show that people who drink tea have lower incidence of certain types of cancer as well. So the health effects of beverages are very well studied. Resveratrol has received a lot of, at the time I started my laboratory, this was in the late 1980s, 90s rather, there had been a lot of reports about resveratrol from red wine. Resveratrol uh, is one of the components of red wine, and it's shown to have many different properties, including antioxidant, anti-inflammatory, uh, anti-cancer, many different properties. So this resveratrol is one of the best studied of the natural products from plants. This is found in grapes, especially among other plants as well, but most notably in grapes and in wine. So that was kind of the impetus for my studies of different fruits. We knew that there were many positive health effects. I was interested in looking at other fruits for their health effects that have been less studied. I'll just mention this is citrus, by the way. This is not one that I was studying, but just a reminder of all the different types of properties. We know some of it from the peel of citrus, certain flavonoids that are known for, as I mentioned, cancer chemoprevention and their impact on the cell cycle, their impact as anti-inflammatory compounds affecting cyclooxygenase enzyme and other ways in which these natural products have been well studied. In my laboratory, we turned to a lesser known plant, which was Jaboticaba. If anyone here is from Brazil, you may be familiar with this. This is a very beloved plant from Brazil, and a lot of the research that's been done on this plant have come from laboratories from Brazil. This is known as Brazilian grape tree. It just has the most interesting, it's called cauliflora. It's actually the fruits are grown directly on the bark. It's just kind of wild when you see the tree in fruit, that these fruits are just kind of studded along the trunk of the tree. It's a very distinct fruit, and it grows pretty rapidly and it matures, and it's these very pleasant tasting fruits that are very nice to eat fresh. They spoil easily. So it's not something you'll see commonly. You can't import them from Brazil because there'd be too much spoilage. So if you ever do see them in your supermarket, they're probably grown in southern Florida. In fact, another famous botanist, and I'll do is David Fairchild. So David Fairchild was a botanist who traveled around the world and was introduced introduced many plants back to southern Florida to the USDA introductory station in southern Florida, and there's actually a botanical garden in southern Florida known as the Fairchild Botanical Garden near Miami that's named after him. And he really, and his home, the Campong, is also in southern, near Miami as well. He introduced Joba de Cava to southern Florida. He said he thought that the climate was very similar to where he collected the Joba de Cava in Brazil. And so he decided that he would bring back the Joba de Cava back to Miami's. And then, despite freezes and hurricanes, it was still resilient, and he was able to maintain this plant in southern Florida. And that's where I did my collections of this plant. Another great park down there is called the Fruit and Spice Park. If you have a chance to go there, I highly recommend that as well. Um, we've published a lot of, we've had a whole range of publications on Joe Boticaba. If you're interested in just a review article, this is a nice place to start where we talk about both the chemistry and the health benefits of Joe Boticaba. You can see that Joe Boticaba, as I mentioned, is mostly fresh, but you can find it in juices and jams and wines. But for the most part, I think most people outside the Brazilian community are not as familiar with this plant. We did kind of classical, what I call natural product or pharmacognosy research, where we did something in what's called as activity guided isolation. So we were really taking the fruits, grinding up the fruits. We had kilograms of fruits that we had collected in Florida. We ground them up, we extracted them with alcohol, and then we did some simple solvent-solvent partition, and then we put them over some columns, and then we basically were able to isolate compounds. And in each step, we tried to test their activity. In this case, it was an antioxidant activity that we were looking at. So we were looking basically for what antioxidants were present in this fruit. This is work that was done by my PhD student, Kurt Reinertsen, and he did a really super job of isolating multiple compounds. And he included one compound that was the first time this compound has ever been described before, and we called it joboticabin. So it's a depside compound up here, and this had very good antioxidant activity. But Kurt went further with this project. He interacted some new collaborations with scientists at Columbia University, or it was literally one of his neighbors that he interacted with, and he found that Janine Darmiento's lab had an interest in anti-inflammatory compounds, specifically for chronic obstructive pulmonary disease. Her lab is interested in compounds that might be useful for the treatment of COPD. And what we found is that we could find some bioactive depsides, and we found that they were able to reduce this inflammatory marker, which is called IL-8. And all of these compounds, but especially compound number one, joboticabin, the new compound, was able to decrease the inflammatory effect. This IL-8 is induced by cigarette smoke, so smokers have higher levels of this IL-8, and as by treating cells in vitro with this compound, joboticabin, we're able to reduce the inflammatory response of the IL-8. So we think this might be a useful drug for the potential treatment of COPD. We did patent this compound, but unfortunately, we haven't gone much further with this compound. I think it still has some promise, but it's one in which we haven't gone further with yet. So overall, I think you know the study of this tropical fruit shows you that you know this this plant is producing antioxidant and anti-inflammatory compounds. Again, these compounds are probably being used to protect the plant from UV stress or for other purposes for the advantage of the plant. But we also, through our studies, found that these compounds could induce inflammatory markers that are associated with COPD, and therefore may be useful drugs for the treatment of COPD. The last study I want to talk about is some of our work on hops. We've worked on hops now for over 20 years. We've worked with a major hop producing company in both in United States and Germany, Hopsteiner. Hopsteiner has been uh, supported our work on hops, and we've worked closely with them for over 20 years on different projects. The project I'm going to talk to you today is really looking at hop chemistry and how hop chemistry can differ based on where a hop plant is grown. So the approach that we use in our laboratory is really the 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 current approach we use is really what I'd say is some of the uh, cutting edge approaches to phytochemistry, which is a metabolomics approach. We're trying to understand, again, these thousands of compounds that are present in a plant. How do we actually get an idea of what they are and how they vary? Uh, you know, the methods we used in the previous two studies were not metabolomics approaches, and so we're able to isolate. We are able to identify just less than 10 compounds, just a handful of compounds using the approach that I showed you in the last two projects. In this project, we're using a metabolomics approach using a a mass spectrometer. This mass spectrometer has great sensitivity, and it also gives us exact mass. We're actually able to identify compounds with great accuracy based on the data that we get from this mass spectrometer. The type of data we're getting is a you know our each of these peaks represents ion abundance. How much of a compound is present under each of these peaks based on how the compounds have ionized, have undergone chemical change within the mass spectrometer. And that's what we're seeing here. We're seeing this, uh, uh, these different compounds that are being eluted from the instrument. And we can start to get a capture of the chemical diversity. And just to give you an idea, each of these peaks that we see here represents at least one different compound. Even these tiny, tiny peaks in the baseline down here, you say, well, they're kind of lost. You can hardly see them down here. Well, they're actually present and they're just in lower abundance, right? So there's, if we were really to count here, we'd see hundreds, if not, you know, thousands of compounds that are present in the hop sphere. And that's all due to these newer technology. I'd say this technology has been with us for the last 20 to 30 years, but it's really been perfected now in the last decade or so. And especially the ways in which we analyze data statistically. So this project, we're really looking at different hop extracts from, um, um, in, in this case, will be from two different locations, and I'm going to go into that study here. The study I'm going to talk about is this one. It's a paper that was done by Tylon Moracle. And Tylon's work was on hops, and he published this paper in Food Chemistry back in 2020. And basically, with the help of our collaborators at Hopsteiner, we got collections of hops from 23 different genotypes from two locations, from Yakima, which is the major hop growing valley in Washington State. This is where a majority of the um, hops that grow in the United States come from the Yakima Valley. And then Kootenai, which is in the mountainous areas, northern part of Washington State, closer to the Canadian border. And this is a higher altitude and in different environments. So we have two different environments, this valley, this Yakima Valley environment, and this more mountainous Kootenai location. So part of the question was, what impact does the environment have on the expression of some of the compounds that really impart a unique flavor? Besides the the alpha acids that are so the bitter compounds, there are highlights and different notes that you get from different hops. And some of those types of flavors include these glycosides. So you can see all these compounds. I'll just call all the compounds on here are different types of glucosides. They have glucose attached, and they impart these different uh, aroma profiles are from these different glucosides. So we have these from the hexanyl glucoside, we have a grassy kind of uh, aroma profile. From this ketone, we have a more raspberry type of aroma that will be, again, it's the hop will still be bitter, but you'll get these notes of raspberry or grassy flavor, or maybe a little floral lavender flavor from linonyl glucoside, or you might get from this other glucoside a more honey or spicy flavor. So we were interested in how these different aroma glucosides would vary based on where the hops grows. This is what we call terroir. That is, that the plant itself is the same, but just the location can impact the flavor. I think people are very aware of terroir from the concept from wines. We know that wines that are grown, even the same grape that's grown in different regions, can have can much different aroma profiles. This is part, the environment, you know, uh, the microbes that are present, um, uh, you know, the rainfall, other things that could impact it, and the altitude, other things that could be impacting the production of these compounds. So this is what we're thinking that will the terroir of the hop change from two different locations? And we used our LCMS um, analysis, and we were able to look at these different cultivars uh, in the two different locations. Okay, so this is the Yakima in red and the Kootenai, the northern collection in blue. And we saw that pretty much statistically, most of the aroma glycosides didn't vary a lot, except for this one that was the hexanyl. This is the grassy, tasty one, right? So maybe not the one we most desired to have overexpressed, but this one seems to be expressed significantly greater in the Yakima Valley than the Kootenai Valley. So I guess if you don't like the grassy flavor, then you could grow your hops up in Kootenai, even though I think is not as you know, uh, that area is not as well known for hop production. But if you are growing it, at least in the season that we tested it for, this hexanyl glucoside will be expressed in much higher levels. So you're going to get a much more grassy taste, hint of grass, let's say a hint of grassy taste to your hops than you would from the um, uh, if it were grown in Kootenai. So that's what we're thinking as far as terroir is concerned. So the genotype of the has a substantial effects on the levels of all of the four aroma glycosides. So going back here, these are the different cultivars. You can see the overall amount of these different glycosides can vary greatly from one here. This is hardly expressed at all to this is expressed at higher amounts. So the genotype has a substantial effect on how much of the aroma glycoside is produced, but the location has a dominant factor of affecting this grassy hexanyl glucoside levels, and this may be due to the insect pressure. So what does that mean for your beer? For your beer, the study suggests that the geographical origin of hops can significantly influence the green or grassy notes in your beer. So for the year that we tested, the Yakima Valley, that with, a lot of those genotypes were expressing higher amounts of this um, hexanyl glucoside. So the beer that will be produced will have a bit more grassy taste to it. So, terroir, I think, is well known in wine, but I think in a lot of different plants and beverages, I guess we can think of coffees and, you know, uh, other beverages that the terroir could have a large impact. Also, the way, obviously, the processing will also have a huge impact on the overall taste of the beverage at the end. I want to end my talk with some thoughts about just botanical drinks. And I think most botanical drinks, contain just maybe 1 to 10 botanicals. That's pretty normal. I don't think we would think that most botanical drinks, if you go into your local supermarket, if you see a juice with, orange and mango, you might think that's kind of crazy. We don't tend to get very complicated mixtures of botanicals in drinks. However, when you think of Chinese medicines, and I do work a lot with Chinese medicines and other projects, those are made up of often 10 to 20 different botanicals. And if you start to think about the chemical complexity, once you introduce 20 different botanicals, we're talking about 20 times as many different natural products that are present potentially. And there are many alcohols. Some of the alcohols that are made, there's a group of alcohols that are made that have up to 40 or even 50 botanicals. Um, and I started thinking about the chemical complexities of these. And the reason I mention this because a few years back, I actually contacted Christine at the Lloyd Library because I was asked by the company that produces Amora Montenegro to give a talk about my research and to tell them, tell them, about their product from my perspective. And when I looked at I was not familiar with their product at all before I gave this talk, and I started to look at beverages with their beverage, which is this Amoro Montenegro. Uh, it contains 40 different botanicals, and I thought that's really crazy. I had not thought about a product containing 40 different botanicals. Um, you know, when I started to look further, I guess Jägermeister actually says they have 56 different botanicals. And this Ouzo from Greece, I think, has about 15 different botanicals that they typically use. I think there are certain gens also that may use more than 10 different botanicals. But I, I can't think of many other examples of botanical, except for Chinese medicines or uh, uh, Indian traditional medicines that really contain so many different botanicals. Uh, but I think if you start to think about it, uh, these mixtures of botanicals, some of these contain plants that are actually, again, on the well known for their flavor, but many of these have important health benefits as well that have been studied over years, as well as, I should say, preservative effects. So some of these, like hops, the original reason hops was used was both for flavor, but also to help preserve the beer as well. And I think we could look at some of these botanicals both in some of these beverages like Montenegro and see that some of them are probably used for flavor, but in addition to flavor, they helped with the preservation of the alcohol as well. I want to say that doing this work, and thanks to Christine, she got me some information as well for this, that, the this particular beverage I thought has a wonderful story to it. It was, I think in the about several hundred years ago, if it wasn't in 16 or 1700s, that uh, this the person who discovered or who produced Montenegro the first time was this Stanislasco Bianchi, who traveled around the world, and he really became impassioned. I would say he was like really an ethnobotanist, someone who was really intrigued by how different cultures use plants. And he gathered plants from around the globe. And I think this is a wonderful example where, traditional Chinese medicine is usually mostly plants that are found in China. This particular botanical beverage was drawing from plants all around the world hundreds of years ago. I think it's such a wonderful thought that this guy went and sourced plants from around the world, put them together into this complex mixture, and he gave what he called a well-balanced flavor, which is these 40 different botanicals. They have only revealed 13. The process is really one of a distillation process. They macerate, they soak them, they distill them, and then they produce their product. They exhibit it. The Lloyd also talks about these different processes in making beverages as well. This is a tried and true method for making beverages. I think what's unique about this product to me is this the number of botanicals and from the different locations and the potential that this could really has the health effects of some of the botanicals that are used here are well known and well characterized for their immunostimulatory effects, their antioxidant effects, and potentially also for their, like the citrus that's present, for their cancer chemopreventive effects as well. Um, so I think it's a fascinating example where there's I, I could only think of a few botanical beverages that really contain so many different botanicals. I'll just end here with just a few future directions where I think the field of beverages is going. I think we're going to continue to see this use of this overlap between health and kind of beverages. I think the idea of adaptogens and other things, we will other ways in which plants, botanical beverages could be healthy or perceived as healthy will be a big push in the market. And also the use of the, I think, multi-botanical, maybe not 40 or 56 botanicals, but I think a greater blend of botanicals will be used. I think also for gut health, we certainly see this now with more and more products that are used like Cambodia for health and gut effects. For immunity and wellness, ginger and turmeric are used in a lot of different products, are used to kind of add to many different beverages as well. And I think there's a great interest in, I think, botanicals. As Lloyd points out in their exhibit, the majority of beverages really are botanically based. And I think we'll just continue to see changes, and as the science becomes better known, I think we'll see different ways in which beverages are kind of blended or produced to make more healthy beverages. There's many great resources. I'll just point out Herbalgram if anyone's interested. It's a monthly magazine that's really well done. It's done by Mark Blumenthal, and I always point my students to this. I'm sorry, it's cut off here, but it's called Herbalgram, and I think Mark does a terrific job in editing this magazine. It's been around for decades. The book that was written, if you're interested in medicinal plants, a book written by my PhD mentors, Walter and Memory Lewis. And also give out a shout out to my former students, Selena and Rachel, who wrote a book, Botany at the Bar, which is a fun read if you're interested in bitters and how plants are used as bitters as well. They did a really nice job in talking about kind of the science behind botany, plants used in making bitters. The work I talked about today was done over the course of 35 years by many, many people. Not all the people who did the work is pictured here. A lot of the work on Jobaticawa was done by Shibiao, work done on also by Kurt Reinertsen as well, did a lot of work, as I mentioned previously. It's really a team of international scientists in my lab, and we're very fortunate to have really wonderful state-of-the-art equipment and really great collaborators from around the world and really great knowledge bases like the Lloyd Library that we depend so much on to be able to do our research. So really a shout out to the Lloyd, and thank you very much for this opportunity to speak to you tonight. And we have a little time for questions. I'm happy to take any if you have any.
Oops. Absolutely. Thank you so much, Ed. That was such a fantastic presentation. Looks like we do have some questions and also some comments as well. Um, First of all, there's a viewer, Kathy. She says that it's really nice to hear you mention Walter and Memory Lewis. She knew them from some herbal medicine conferences, and she still refers to her to their research in her classes, and also saying that Jeboticaba, if I'm pronouncing that correctly, is
Maybe better
abundant
than I do. Thank you.
is abundant on the Big Island in Hawaii, and
Oh,
that
great.
it's oftentimes sold at fruit markets there. Um, and I also just want to say that we at the Lloyd do have a subscription to Herbalgram, and we also do have Walter Memory Lewis's conference proceedings and books here. So come on down and take a look at their research. So, first question for you, Ed, is from Maria. She would like to know if do pollinators and other plants in the vicinity play a role in hop aroma's diversity? Was that also part of the study as well?
Let me think if I can remember my hop. And is hop when pollinated? Let me think about this for a second. I can't remember the pollination of hop. I'm thinking that they are dioecious. They do have the male and female. And how are they pollinated? I'm sorry, I don't remember that specifically. So I don't think I could really answer that question in detail. But uh, um, it's yeah, I so know if it's hop. I think it's. Thinking it's wind pollinated, but I'm not positive about that. So I better not speak anymore.
Okay. Um, well, next question we have is from Bill, and he would like to know if is the amount of research devoted to health effects of plants increasing significantly?
The amount of research devoted to health impact, I think absolutely. I think especially in China, I do a lot of work in China, and they've invested greatly in the most modern of methods to study mostly traditional medicine, but they go beyond their traditional medicines as well. But there's been but I think you know and I think there's been I think as far as in the United States, I think it's mostly within kind of the health effects are there is part of NIH that looks at uh, the Center for Complementary and Integrative Medicine Health, and CCIH looks at a lot of botanicals as well. And I think I saw one of my former students, Ella Vardaman, I'll give you a shout out, who was supported by, and, oh, yes, she is there. She was supported by NIH, but I'd say that's a very small part of NIH, research portfolio is botanicals, but it's, for certain areas, they do it. They do have research involved in it. I'd say a lot of the research, that you'll see done on botanicals, a lot of it is coming from China and some other countries that have invested heavily in kind of botanicals. And I'd say a lot of countries, World Health Organization, shows a lot of countries that don't have resources to purchase, expensive drugs. Really, a lot of countries rely on traditional medicines. So even smaller countries. Currently, we have a researcher from Burkina Faso in my laboratory. He's very interested in the health effects of medicinal plants from his country, and I think it's the countries that you know you know really often turn to their own traditional medicines first. So the more knowledge we can have about that, you say, well, why do we need it if we have these great antibiotics? Well, a lot of people can't afford to get these modern medicines and really have to rely on the traditional medicines. And many of these are very effective, but less studied.
Bill has a follow-up question to asking, are medical schools devoting more time to herbal medicine study?
There had been a push, and the part of my work at Columbia University, this was 20 years ago, they had a Center for Complementary and Alternative Medicine. It was called the Rosenthal Center. It was Freddie Cronenberg, who's published a lot, especially on black cohosh. A lot of our work on black cohosh was with that. And she had, at that time, had a very large grant from NIH to look at that. So I think often there'll be a small module of the kind of medical education that's done it, and there may be some group within a medical school that has some interest uh, in botanicals, the medicinal properties of botanicals. But I'd still say it's a small group. I think but there is at least a recognition of it. especially, and there's been some significant funding across the United States. The NIH has sponsored these botanical centers. And so there's one at there has been one previously at at Michigan, I'm not sorry, at Mississippi, University of Mississippi at Oxford has had a botanical center for a long time. And NIH has really kind of supported a small number of botanical centers across the United States that have focused uh, on natural products.
Um, I have just a question or two that I came up with as I was listening, but when you were talking about how the different locations and different conditions affect the different types of hops and grapes, is that a common observation that you see in other plants in your study? And also, does the environment really impact those compounds? Because it made me think about how the wildfires that happened in California for the past few summers or so have really impacted the wine industry and the grapes, and therefore leading to different qualities of the wine product.
Yeah, absolutely. And that's a fascinating example. One of my colleagues who actually is a chemist but also grows grapes, he had he actually published a paper about this because his vineyard was, I think he tainted basically by the smoke, so we had this problem where the compounds from the burning of the wood would actually taint the grapes. So I guess that's kind of a different issue than terroir, which is more about the growing conditions. I guess you could consider smoke as a part of the growing conditions because you might consider water, kind of water stress could be part of that. The microbiome, the plants, the microorganisms that are associated with the roots of the plant could impact the production of compounds as well. The altitude, all these could have an impact. And so I think this concept of terroir, you said, definitely the grapes, and now we've studied in hops, although I think we were kind of the first to really use the word terroir with hops. I think there are many different plants that would have it. When I was giving my talk, I was thinking about coffee, and I think certainly the different coffee, but I think coffee is also something that's really heavily processed too, and tea as well. You talked about that in your exhibit too, about the different oxidation of tea, and so that has such an impact also on the flavor of the tea. Some of these may actually kind of override some, those subtle changes with the hop glycosides where you get a little grassier taste. You know, through processing, you may lose some of that as well. So you may not even, we may be able to pick it up chemically, but actually when you taste it, it may be harder for us to taste those. So I think this area of terroir is still being, people are still interested in this for sure. And I think it definitely goes beyond just, grapes.
Sure. And one final follow-up question I have is the age of the plant. Are you for, well, I guess with any example that you've used, do you focus on, an early bud of the plant, or do you wait for it to fully mature or towards the end of its life? When are you guys looking at the compounds
Right.
typically?
And that would make a huge change in the chemistry, right? So what you're suggesting is absolutely true that, I mean, immature leaf versus a mature tea leaf, the chemistry will be completely different. So I think you're definitely onto something. What we try to do is how people use them,
Mhm.
So we try to think, if so we try to get the mature Joba de Cava. We don't just get it when it starts to, just starts to fruit. We wait until it's actually when people would actually eat it. That can be tricky when you're in the field and not always possible, but that's for the most part we try to harvest when people typically would harvest. For tea, for example, you have to harvest in a very specific way because there are certain enzymes that will continue to alter the chemistry once you pick the tea leaf. So you have to think about carefully about post-harvest impact of some of the chemistry that will happen even after the plant is harvested. So
Sounds like ethnobotany is a real part of your
It's a
research, which
driver. It's
is,
a driver of
yes.
all this research is ethnobotany. I really would like to make, and I was glad to hear about Walter Memory Lewis. Walter's passed away, by the way, my PhD mentor, but his wife Memory is 93, and
Oh,
we just traveled to China a few years ago for her first trip to China. I took her, and she joined me in China, so she's still very active. So the person who said that they knew her, I was glad to hear that, and I think ethnobotany for us is really a foundation. It's what we stand on. We wouldn't do any of these studies except for our knowledge from humans of using these plants.
thank you. Thank you very much, Ed. And I don't see any more questions. So, with that, our time here has come to a close. So, I want to thank Ed once again for this wonderful and insightful lecture. And thank you to folks too who sent in comments and questions as well. I must also thank the members of the Lloyd Library Museum who make programs like these possible through their memberships. If you would like to learn more about our membership programs, you can click this link that is in the chat, or you can visit our website at www.lloydlibrary.org. And then, if you would also like to see more lectures and programs like this, you can check out our YouTube channel, which you can find a link to that also on our website, or you can search for youtube.com/at Lloyd Library. And so once again, thank you everybody. Thank you, Ed. I wish you all good night and to see you soon at the Lloyd.
Thank
Thank
you,
you.
Christine.